Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers in preprints in science. Before we dive in, if you enjoy what we do, please take a second to like and subscribe. It really helps support the Sand Science Experience. Okay, so today we're peeling back the layers on something pretty fascinating in aging research. It's a new tool called EnsembleAge. And our mission really is to figure out how this thing is changing the game for measuring biological age. You know, how well interventions are working to slow things down, maybe even reverse aging. This isn't just counting birthdays. It's about getting a real look inside, the molecular level, and how that could genuinely shape the future of health and longevity for you. So let's start there. When we talk about measuring biological age, what's been the biggest headache for researchers? Why is it so tricky?
Yeah, that's a really good place to start. A huge challenge has just been the inconsistency of the tools we've had, these things called epigenetic clocks.
Right, those estimate age based on DNA methylation.
Exactly, those little chemical tags on DNA that change as we age, affecting our genes. But the problem was you could run different clocks on the same sample and, well, get different answers.
Ah, so conflicting results. Makes it tough to know if something's actually working, I imagine.
Incredibly tough. You couldn't be confident in evaluating interventions. So that's where EnsembleAge comes in. It tackles that limitation head on.
Okay, so how does it do that? What makes it different? Is it just a better clock?
It's actually not just one clock. That's the key. It's a suite of ensemble-based epigenetic clocks. Think of ensemble like getting a consensus opinion from a panel of experts instead of just one voice.
Okay, like combining strengths.
Precisely. It gives a much more robust and, well, reliable assessment. And they built this using a really extensive data set called MethylGauge.
MethylGauge?
Yeah, think of it like a huge training ground. It compiled DNA methylation data from, I think, over 200 controlled experiments in mice. Yeah, involving all sorts of things, stuff that accelerates aging, like stress, and things meant to be rejuvenating across different tissues, too. It really let EnsembleAge learn the subtle signals of aging.
Okay, that sounds promising on paper, but the proof is in the pudding, right? How did it actually perform when they tested it against the older clocks?
Yeah.
Did it really, deliver?
It absolutely did. The empirical tests, the evaluations, they showed EnsembleAge significantly outperforms the older clocks for detecting both the bad stuff, pro-aging changes, and the good stuff, rejuvenation.
Better how? More sensitive.
Much more sensitive, yeah. And crucially, lower false positives and false negatives, so it's more accurate overall.
Can you give us an example, something concrete?
Sure. Take Huntington's disease mouse models. EnsembleAge picked up clear signs of accelerated aging in those mice.
Okay.
And here's the kicker. It found those signs even in brain regions like the cerebral cortex and striatum, places where the older clocks just drew a blank. They couldn't detect any significant aging changes there before.
Wow, okay. That is sensitive. What about the other side? The rejuvenation part, that's what everyone's excited about.
Right, well, it showed superior sensitivity there, too. It successfully detected the anti-aging effects of rapamycin treatment.
Which other clocks missed?
Which many of the original clocks completely failed to capture, yeah. And maybe a bit chillingly, it also connected faster epigenetic aging in mouse tail samples directly to dying earlier, so a real link to mortality.
Okay, those are strong results in mice. But, the big hurdle is always translating mouse findings to humans. It's notoriously difficult. What makes this EnsembleAge human mouse thing potentially better for that?
That's the critical question, absolutely. So they specifically developed this extension, EnsembleAge human mouse, for cross-species analysis.
How does that help?
Well, it focuses on DNA methylation sites that are conserved through evolution. The bits of the code, the methylation patterns that are fundamentally similar between mice and humans.
Ah, so it's comparing apples to apples, epigenetically speaking.
Kind of, yeah. It makes the findings from mice much more likely to be relevant, more readily applicable to human studies.
And why is it so much more accurate, fundamentally? What's it picking up on?
It seems to be zeroing in on methylation changes in really crucial spots. These key regulatory regions like promoter regions and 5-UTRs, think of them as the dimmer switches or main controls for genes, and within CPG islands too.
Okay.
And it also connects these changes to specific ways our DNA is packaged, these chromatin states that have already been linked to how long mammals can live, like states called TSS1, TSS2, PROMF5. It suggests EnsembleAge is tapping into something deeper about biological longevity, not just counting years.
So let's bring back, what does all all this mean for you listening right now? This isn't just some obscure finding for lab researchers. It feels like a big step towards actually understanding human biological age much more accurately.
I mean, imagine the potential for precision medicine.
No more guesswork, perhaps?
Yeah.
Like getting a personalized biological age report card that shows how your diet, your exercise, maybe a new therapy is really affecting your aging inside.
That's the vision, allowing for real-time adjustments based on a much clearer epigenetic picture. It really underscores that precise measurement is just fundamental. You can't improve what you can't accurately measure.
Right. Knowledge starts with measurement.
It does. And maybe the provocative thought to leave with is this. As tools like EnsembleAge get even better, more refined, how might our power to understand, track, and maybe even intervene in aging truly change our whole approach to health, to disease in the years to come? It could be quite transformative.
A profound question indeed. Well, that's all the time we have for this deep dive. A huge thank you for exploring EnsembleAge with us. Remember to like this video and subscribe to the channel if you found this interesting. It really helps us keep bringing you content from the ScienceScience experience. We'll see you next time.